Hey everyone, and a very warm welcome to Cognito Key. Today, we're going to dive into a really fascinating biological process. It's a simple clumping reaction that unlocks a ton of information, from our own blood type to viruses we can't even see. So, have you ever wondered how a single drop of blood can tell us so much? How it can instantly reveal your blood type, or even help us count millions of invisible viruses? Well, the answer lies in one elegant, visible reaction. In this explainer, we're going to break down the science of hemagglutination, what it is, how it works, and why this simple clumping is so incredibly important. Okay, let's get right into it.
All right, first things first, let's get to the heart of it and really understand the basic principle behind this whole phenomenon.
So what exactly is hemagglutination? To put it simply, it's a reaction where red blood cells, also known as erythrocytes, clump together. And the best part? This clumping is something we can actually see with our own eyes, which makes it an unbelievably useful tool for diagnostics. To really get it, let's just break the word down. It makes it super easy to remember. The first part, heme, that just refers to the blood component we're focusing on, the red blood cells. And the second part, agglutination, is really just a scientific way of saying an aggregation or a clumping of particles. So you put them together, and what do you get? The clumping of red blood cells. See? Simple, right?
But what actually makes them clump up like that? Well, the reaction is caused by agents that are called hemagglutinins. You can just think of them as a kind of molecular glue. Some common examples you probably heard of are antibodies from our immune system or even the glycoproteins you find on the surface of viruses. When these agents are around, they literally grab onto multiple red blood cells and make them stick together in a visible cluster.
Now let's see how this simple clumping is used in one of the most common and vital medical tests out there, finding out your blood type. Getting this right is quite literally a matter of life and death when it comes to blood transfusions. So here's the crucial point. Your red blood cells have these tiny little markers on their surface called antigens. When these antigens meet their specific matching antibody, they bind together. And that's what triggers the clumping reaction we've been talking about. This simple interaction is the key to everything.
Okay, let's walk through this chart. Imagine a lab tech mixes a drop of your blood with two different solutions. One is antiserum A, and the other is antiserum B. Now, if your blood clumps up with antiserum A, it means you have A antigens, so your blood type is A. If it clumps with antiserum B, you're type B. What if it clumps with both? You're type AB. And if there's no clumping at all, then you're the universal donor type O. It's that crystal clear visual confirmation that makes the test so reliable.
But you know, blood typing is just one application. Hemagglutination also gives us this brilliant way to quantify things that are far too small for us to see, like viruses.
This is done using something called a viral hemagglutination assay. Here's how it works in 3 really simple steps. First, a virus sample is diluted over and over again across a special plate. Second, a standard amount of red blood cells is added to every single well on that plate. And finally, you just watch and wait. You see, certain viruses, like influenza, are natural hemagglutinins. They will latch onto the red blood cells and cause them to clump up.
And this just illustrates the results so beautifully. If the virus is present, it forces the red blood cells to form this sort of lattice network, creating a diffused mat all across the bottom of the well. But if there's no virus, the red blood cells have nothing to stick to, so they just settle down into a neat little dot, or a pellet at the bottom. A mat means virus, a dot means no virus. It's a wonderfully simple visual readout.
Now, of course, like any scientific technique, this assay has its strengths and, naturally, its weaknesses. So let's take a quick, balanced look at both. The advantages are pretty clear. It's a simple, reliable, and relatively cheap test to run. Plus, it's fast. You can get your results in just a few hours, which is absolutely fantastic for diagnostics. This is why it's a go-to tool in so many labs all over the world.
However, it does have its limitations. This isn't a foolproof push-a-button kind of test. It actually requires a good amount of technical skill to perform correctly, because things like timing and concentrations have to be absolutely precise. If it's not handled carefully, other factors can sometimes interfere and give you the wrong results. So let's zoom out for a second and look at the big picture. Why does this simple clumping reaction truly matter in the grand scheme of things?
So why does it matter? Because as we've seen, this one single technique transformed at least 2 critical fields. First, it gave us a fast, reliable way to determine blood types, which is essential for safe transfusions.
And second, it provided a simple method for counting viruses, which is vital for things like developing the annual flu vaccine.
And that is the real takeaway here. Haemagglutination is this elegant, beautifully simple concept. It's a visible clumping that allows us to see and understand a whole world of microscopic information that would otherwise stay hidden from us.
And that kind of leaves us with a final thought, doesn't it? If a process as straightforward as watching cells clump together can tell us so much, what other profound biological secrets are just waiting out there to be unlocked by the next simple, elegant test? Thank you so much for joining us on Cognito Key for this explainer. If you found it helpful, please do like, share, and subscribe for more breakdowns of complex topics. We'll see you in the next one.